Hello, this is Gene Trowers from the University of Arizona, and today's topic concerns stimulation and inhibition of gastric acid. There are three main factors involved in the stimulation of stomach acid. One would be from the vagus nerve, and it will release acetylcholine, and this will be during its direct stimulatory process. Now, the vagus nerve also has an indirect process. We'll say a little bit more about that shortly. So, via the direct process, vagus nerve releases acetylcholine, which is a neurotransmitter, and it will stimulate a muscarinic receptor, M3, on the basal portion of the parietal cell. That's the portion of the cell that faces the blood. Now, once this muscarinic receptor has been stimulated, it in turn will stimulate what we'll call a G sub Q protein and then act through a secondary messenger, inositol 1,4,5 triphosphate, say that 3 times rapidly, as well as intracellular calcium. And that secondary messenger will then stimulate this hydrogen potassium ATPase proton pump to secrete hydrogen ion and ultimately hydrochloric acid. Now, the vagus nerve, as I mentioned, can stimulate gastric acid secretion by an indirect process. The vagus nerve, its terminal portion, releases GRP, gastrin-releasing peptide. And that will stimulate the G cells to release gastrin. As you recall from your bio days, gastrin is stimulated by the presence of food in the stomach, stretch in your stomach, especially small peptides in the stomach. And so the gastrin, which will act as a hormone, will stimulate this cholecystokinin B, CCKB receptor, and it will follow this similar stimulatory cascade that we outlined for the vagus nerve direct stimulatory pathway. So that was our second major stimulatory factor for gastric acid secretion. And the third major factor arises from the enterochromaffin-like cells, or the ECL cells. Now, the ECL cells will sit near the parietal cells in the stomach region. And the ECL cells will release a paracrine substance called histamine. So this histamine will travel a short distance to the parietal cell and stimulate the H2 receptor on the parietal cell. This in turn will lead to the increase in intracellular cyclic AMP, which will then stimulate acid secretion via this hydrogen potassium proton pump factory. Okay, so as you can see, histamine acts via a different stimulatory pathway compared to what would take place with acid stimulation due to acetylcholine or gastrin. And this can have important ramifications. We'll say a little bit more about that. At this point, I'd like to bring up the concept of potentiation. You know, potentiation occurs when you simultaneously stimulate two stimulatory factors and you get a response. Now, if you were to take each individual stimulatory factor stimulated and then add those two responses, when you undergo potentiation, the response that you get is so much greater than what you would have gotten if you had simply relied upon adding those two individual stimuli. So, if you were to give a patient uh, histamine, then this would stimulate acid secretion, but it also potentiates the effects of gastrin as well as acetylcholine. And in a similar way, if you give a patient acetylcholine, it will have a potentiating effect on gastrin's stimulatory effect as well as histamine's stimulatory effect. Now that's all well and good, but how does this play in everyday life or when you go to the doctors? Okay, patient has a problem with increased acid. Well, let's say we gave them a medication, atropine. Okay, now atropine is what we call a muscarinic blocker. Fancy word. But basically what it does, it blocks the stimulation of acetylcholine at this M3 muscarinic receptor on the parietal cell, and it inhibits this cascade of acid stimulation. Okay? Now, what about if we give the patient a different classification of medication called a histamine receptor antagonist? Now, this is an agent similar to Tagamet or cimetidine that you can purchase at the store. This would block histamine stimulation of the H2 receptor and its cascading effect on acid stimulation. Okay? Now, what would happen if we gave the patient what we call a proton pump inhibitor, PPI? Now, this is an agent such as omeprazole or Prilosec, the famous purple pill. And you could think of it, this proton pump inhibitor, as a smart bomb. It's a direct hit on that proton producing factory, and that proton secretion leads to the secretion of hydrochloric acid within the gastric lumen. Now, there are some other mechanisms of acid suppression within the body. There are agents such as somatostatin, which are produced by D cells, and there are other agents which can cause acid inhibition, such as prostaglandins. Probably a topic for another day, but needless to say, these two agents act via what we would call a G sub I protein that has an inhibitory effect on intracellular cyclic AMP and hence significantly reduce or block acid production. So in review, we talked about the three main stimulatory factors for acid secretion in the stomach: acetylcholine, the effect of gastrin, the effect of histamine. And we also outlined the consequences of if we give a patient certain types of medication, such as atropine, where it blocks at this muscarinic receptor, or if we give a patient HRA, histamine receptor antagonist, blocks at the level of an H2 receptor, and these will be agents such as cimetidine or Tagamet. And then also, we talked about if we gave a patient the purple pill, this proton pump inhibitor, Prilosec or omeprazole, its effect at the level of the proton pump. Hope that this increased your understanding of the stimulation and inhibition of gastric acid secretion. Thanks so much. Have a great day.